So, welcome to part two of our immune notes. We talked last class about how our specific immunity in our body is created by our lymphocytes. And we have two types of lymphocytes, our T lymphocytes and B lymphocytes. Our T cells are formed or differentiated in the thymus gland, whereas our B cells it's sort of unknown, but you could probably go with B for bone marrow. So our B cells are differentiated in the bone marrow. And those are stored in a variety of places. And those hang out and wait and try to catch any antigen-bearing agents, so foreign substances that enter our body, and then they have different actions. Our T cells act by interacting directly, called cell-mediated immunity, and our B cells are going to act indirectly by producing and secreting antibodies. And B cells will then travel through the body's fluids and will encounter whatever that foreign antigen-bearing cell is. And this is called antibody-mediated immunity, or AMI. So within our body, there are clones of lymphocytes within the populations of our T cells and B cells of each person, and then there's millions of different varieties of those lymphocytes. Now, the members of each variety are alike, and they have a particular type of antigen receptor on their surface membrane. That responds only to a specific antigen. So it's kind of like puzzle pieces, right? So we always show the picture of the puzzle piece concept that each B cell would fit a certain type of a pathogen, and it's kind of like a puzzle. So how do we activate the B cells? The B cells are activated when they encounter an antigen whose molecular shape fits the shape of the B cell's antigen receptor. So that looks a little something like this. So we have a B cell, it has a particular antigen, a receptor site on each different B cell. So this one has like a little triangle receptor site. This one has like more of a little arrow receptor site sticking out. So if this cell is the antigen right here, and the B cell combines with it, it creates an antigen receptor combination, which then would trigger or activate that B cell. And an activated B cell would then enlarge its clone or make lots more of itself. So back to our notes. So in response to the receptor antigen combo, the B cell undergoes mitosis and its clone is enlarged. So it would make a whole bunch more of itself. So some of the newly formed members of the clone will then become what are called plasma cells. And they then make use of their DNA to produce antibodies. So antibodies are proteins called immunoglobulins. So we talked a little about immunoglobulins as the I that we use in blood typing. So if you look at an antibody molecule, they have some variable regions and some constant regions. So in this picture, there are constant heavy chains. Basically, let me back up. The antibody molecule is made up of 4 chains of amino acids. And those are linked by disulfide bonds. So we see the disulfide bonds here that are holding together. Here's 1 chain, 2 chains, 3. These are a continuous chain here. So 1 chain, 2 chains, 3 chains, and 4 chains. So, and then there's constant regions and variable regions. The variable region is what is going to be able to attach to the antigen receptor site. So look at the key here. Oops. Move my picture. Don't move my picture. Okay, this is tricky. Okay, everybody laugh at Gorka. All right, so there's the key. So the variable region is the light blue. The dark blue is the constant region of the light chain, and then there's the variable region of the heavy chain, which is the light purple, and then the constant region of the heavy chain. So if you haven't already, go ahead and draw a little antibody on your paper. And so we've got our 2 heavy chains, or 2 big chains, and then 2 light chains, and they're held together with the disulfide bonds. I'm still here, just letting you draw. So 2 heavy chains, 2 light chains, variable regions, and constant regions. And if you didn't catch that, it is in your book. So antibodies are proteins called immunoglobulins. And each is composed of 4 chains of amino acids. That are linked by disulfide bonds. The variable portion of the chain are specialized to react with the shape of the antigen molecule. So that's where we get that concept of the different shapes of the antibodies. So they each have, in that variable region, have the ability to attach to different antigens. So continuing to our next page here.